Hello student, how are you? Welcome once again in our SAS Academy, our YouTube channel SAS Academy. Our contact number 9163398358. Please subscribe our channel. Earlier video we discussed about how to crack NEET and what are the important topics of the NEET exam in biology. What a very important chapter we discussed about previous video. In this video we discuss about very important topic not only for NEET student but also the class 12 board and also class 10 board student, that is principle of inheritance. What are the principle and what is inheritance? First of all, we know about what is inheritance. Inheritance means that is transmission of character from parents to offspring. That is transmission of character. Transmission of character from parents to offspring. Then first of all we know about what is character. That is the morphological expression of the organism is called character. That is traits. What is traits? Traits means the alternating form of a character. Alternating form of a character, this is called traits. That is the height of the stem, this is the character. They have two alternative form, either tall or dwarf. Color of the seed, this is also either yellow or green. This is the two alternative form. And then we know about this character are controlled by very important things. This character is controlled by part of the DNA which consists of many of nucleotides. This part of the DNA is called gene. And this gene also have a two alternating form of a gene. This is called allele or allelomorph. That is one gene. That is a T is a gene. They have the two alternating form. The tallness is the character. They are controlled by a gene. They have the two alternating form. These are gene, two alternating form. One allele, capital T, another allele, the small t. That is, allele means when the homologous chromosomes at the particular region of the homologous chromosome, this particular region is called locus. This locus region, there are two alternating form or contrasting form. These two contrasting form, either capital T or small t, each of this contrasting form of a gene, this is called allele. This is called allele. Now we discuss about another two main terminology, that is genotype and phenotype. Genotype means gene constitute, the genetic constitute of an organism. This is called genotype. And morphological expression of an organism morphological expression of an organism, this is called phenotype. Then we also know about in this chapter another two main terminology. One is the homozygous, another is the heterozygous. Homozygous means when the two allele are same, that is capital T or small t. Homozygous also, this is the pure form. And on the other hand, the heterozygous, there are two form of allele are present. These are hybrid trait. This is all about main terminology in principle of inheritance. First, the concept of gene put forward by Gregor Johann Mendel. This is first the gene concept, or first this gene concept they express as a factor. This factor are controlled by the character, and this character is controlled by this factor. This gene or factor first introduced by their renowned experiment in garden pea or Pisum sativum. This scientist is Gregor Johann Mendel. All the gene concept, all their inheritance characters are put forward by the renowned scientist Gregor Johann Mendel. That's why the Mendel also called, that is the father of genetics. And they put forward two main experiments. One is the monohybrid cross or hybridization, and dihybrid cross or dihybrid hybridization. Monohybrid cross, here the two parents are concerned about one pair of trait or one character, that is tall and dwarf. This is the one pair of trait concerned about in this cross, and when that is capital T, small t. On the other hand, the dihybrid cross, the two parent, the concern about two pair of traits or two characters are involved in this cross, that is capital R for round and capital Y, that is yellow, and small r, this is the wrinkle, small y is the green. That is the color of the seed is a character. There are two forms, round and wrinkle. This is the one pair of trait, and the shape of the seed. This is the two traits. One is the round, another is the wrinkle. And color of the seed, this is the yellow and the green. There is the two traits. In this chapter, we know about how these monohybrid cross are done in Pisum sativum by Mendel. Mendel first, they took the two pure pea plant. One is the pure tall, that is capital T, and another is the pure dwarf, small t. This is the P generation, parental generation. Then they produce gamete. The capital, always the tall produce one type of gamete because the homozygous state. On the other hand, dwarf also produce one type of gamete, that is small t gamete. These two one are fused or fertilization to form the zygote and ultimately to form the F1 first filial generations are produced, these are morphologically tall, or that is phenotypically they are tall, but genotypically these are hybrid or heterozygous. Then Mendel crossed these two hybrid pea plant, hybrid tall plant, capital T, small t, capital T, small t. And then F2 second filial generation, that is two gametes are produced, capital T, small t, capital T, small t. There are some rules. 1t always the multiply with the 2. First capital T multiply with tt, capital T, small t. Always there are very important thing, that is in character height of the stem there are two alternating allele. One is the tall, another is the dwarf. Tall arise in F1, that's why tall character is the dominant character. On the other hand, the dwarf character, dwarf next, this trait is the recessive character or recessive trait. That's why always when you write this thing heterozygous condition, always dominant allele are write first, recessive allele write second. Then capital T, small t, small t, small t. Ultimately, that is, this is the tall plant, this is the pure tall, this is the hybrid tall, this is the hybrid tall and pure dwarf plant. In this regard, very important thing, that is what is the phenotypic and genotypic ratio of this monohybrid cross. That is phenotypic ratio that is tall monohybrid cross, they put forward a law of segregation. In this law we stated that the allele for each character, they existing in pair in an organism, they do never blend, they segregated from each other during formation of gamete. That is, the capital T, small t, they produce capital T gamete and they produce the small t gamete. When they are fertilized to form capital T, small t, this is heterozygous. Here F1, the tall character is expressed, but the dwarf character cannot blend it. This allele of tallness and allele of dwarfness, they are segregated from each other during the formation of gamete. That's why in F2 we produce one pure tall plant, two hybrid tall plant and one pure dwarf plant. This is all about the law of segregation, and further video we discuss about dihybrid cross, also the deviation of the Mendel law, that is very important law. The incomplete dominance, that is multiple allelism, co-dominance, pleiotropy, polygenic inheritance, all are discussed later video. So please, student request to you, please subscribe our SAS Academy, and any doubt your mind, please contact our contact number is 9163 398 358. So in our also Facebook page available, SAS Academy. So student, please join us with our YouTube video. Thank you so much. Good night.